one of my experience. I have been done my practical during my on my internship at Zoom Malacca, where one of the it's like the conservation that have been done from the zoo, from the captivity, is try to like, it's not rematching, like we try to make sorry, back again. Sorry. Try to match. Mean, like, we try to do the breeding in the captivity itself. Like, if I'm not mistaken, tapir at the Zoo Malacca is actually a male one, named Tuah, if I'm not mistaken. Then we try to invite another tapir. If I'm not mistaken, it's from Pahang. And then we try to match it, and I don't know whether it's how the process will have been done. Something like, maybe I think it works, but I don't know whether the female apa kalau we're expecting like maybe the female already been pregnant or something like that lah. I don't know whether it's they're achieving their goals to do the conservation in the captivity. So I just want to share my experience in the captivity. That's all. Thank you.
Can you hear me? Here in this room, can you hear me? Unclear. Let me just try now. Can you hear me better now? Oh, can you hear me? No, not at all. And how about now? Yeah. All right. Okay. Sorry, everyone. I think it's good if you select one coordinator here first, a facilitator that would sort of guide you through the system, so to say, so that we manage to compile all the different suggestions. I mean, and anything goes. It's open floor, so any suggestions would be good. So somebody would have to be a note taker and take it down, and then another person would have to sort of drive the process. And sorry, it can't be me because I'm going to jump into the other room soon, and vice versa. And anyone wants to volunteer on this? Raise your hand, maybe.
So good afternoon. Is there a checklist that we need to go through? Because right now I think most of us are not sure what we need to
do. Thank you, Sandie. Good to see you again.
Yeah.
Well, there's not a checklist per se, so because we don't really have a starting place today. It's not really a workshop, but it's just sort of formulating some ideas that we can work on later on, hopefully in a proper few-day workshop. So So it's just some ideas, what you feel from an ex-situ perspective, what can we do better for Malaysian tapir conservation or the regional tapir conservation? From an ex-situ, do we need to be better at cryobanking or sort of secure the future? What about artificial insemination and these kind of things?
Okay. Yeah. So I guess we'd like to get feedback from Donny because as you know, I've left the water department for some time. So what is the current situation they have? Do they have a biobank? For what I know, Sungai Dusun is still operating. I think they're still actively breeding the animals. So I guess what you're asking is what's the next step we want to take this forward, right?
Yes, absolutely. But it could also be on some of the universities or NGOs that feel, look, we have some new tech or better tech or education that we can pass on, or work with the Philippines in Malaysia or in Thailand and doing a lot of the DNA work there, for example. So it might be something like that at the university level.
Okay. So currently I'm at the University of Malaysia Kelantan. We are very new, but we are willing to help where possible. I think a few times we went down to Sungai to try to help with the Malayan tiger breeding. I'm also on the board of the Asian Society of Conservation Medicine. So I think over there we'll get quite a lot of resources. We can find expertise in the Asian level. What else you can do? Donny, what do you have in your mind?
Yeah, hi everyone.
Hang on, Donny. Can I just ask anybody keeping notes on this, then taking down some? Anyone volunteer to do that?
Yeah, we need somebody to jot down what we are discussing. Yeah.
I mean, not everything you say, but the proposals and suggestions.
Yeah.
Here, I'll do Mark from UPM.
Okay, thank you.
Great, thank you, Mark.
Yeah.
All right. Maybe I can give some insight on the ex-situ breeding programs in Perhilitan. As mentioned by Dr. David in the country report, we have few zoos and one conservation center under Perhilitan, which involved in the breeding of Malayan tapir. Currently, we have around 40 Malayan tapir, which is in captivity, which most of it involved in the breeding of Malayan tapir in captivity. So for Sungai Dusun Wildlife Conservation Center, for the past 17 years, we already have 19 calf bred in that center. However, we could enhance or increase the reproductions or breeding rate of the Malayan tapir in captivity via involvement of assisted reproductive technology, which I think including more advanced reproductive technology like artificial inseminations, embryo transfer, and so on. For the time being, what we do involving with ART or assisted reproductive technology include semen collections, what else? Evaluations, and some cryopreservations. But we couldn't do further, as we need to understand their cycles, which is to understand their estrous cycle. We have research ongoing, which will be completed soon, and we might need to do further research on artificial inseminations and other advanced reproductive technology. That is on breeding side. But we also need to embark or to do more research on the rewilding of the Malayan tapir, especially those bred in captivity. So those insight that I can share for the time being. Yeah.
Is the department looking out for new techniques or new technology, or how can we help?
All right. As mentioned by Dr. Zainal, that learning from the Sumatran rhinoceros, in which when we are in crisis, we are not ready. So I think universities can involve in preparing teams of what you call it, especially in reproductions of the species. In future, when the needs arise, in which when we are in crisis, we already have the expert team in place to do the works. Yeah. So I think the works to prepare the teams should be started from now, and the department need collaborations from universities and also from the regions. I mean, from the ASEAN countries.
Okay, I think we can, if from department initiate some sort of meeting or some sort, we can bring people in to start that. But currently, you say just now there's about 40 plus animals, right, in captivity? Throughout.
Yeah, we have about 41 heads of Malayan Tapir captivity, but not all involved in the breeding of in breedings. Yeah, in reproductions, as some old, some is not viable for reproductions, and some is injured animals, and some is still at very young age. Yeah, but I would say half of it they involved in the breeding of in captivity. Yeah.
I'm just thinking, are we reaching the maximum capacity for the captive population? Because like tapir, it's not a cattle. You can't put all many of them together. They need individual housing. So
yeah, I think
yeah, like they do like we want a lot, but where to put them?
I think first in Sungai Dusun Wildlife Conservation Center, I'm not sure in zoo, but in Sungai Dusun Conservation Center, I think that is our problems. We are successful in breeding the tapir naturally in captivity, but we have a very limited space to accommodate them. So we are thinking of rewilding them, but if we can rewild them, do we it is necessary to release them to the wild, since in the wild still have viable populations as mentioned by Dr. Karl, but in fact we need to study the what you call it, the rewilding process. Yeah, we don't really have the rewilding SOP at the moment, but I think it's possible to be done. But one other things that we can be done is to collaborate or to link with other international institutions like zoos and other zoological parks internationally, so we can reproduce and we can exchange with that institutions. Yeah. So I mean, we produce and to exchange with or to send to other zoological parks that are in need of Malayan tapir. Yeah.
Because we don't want to hit the same problem we did with the Malayan tigers one time, like 20 plus in the zoo, and We
Yes.
can't breed, we can't do anything, we can't send them out. It's like we are caught with ourselves. You know, what to do.
Yes, but in that case,
Yeah.
maybe it's beyond my say. I mean, I have no say on this on
Yeah,
policy.
yeah, obviously, yeah. But
yeah,
I mean, we have to think about that.
yeah.
If we have a limitation in terms of to keeping them captivity, and to a point we have to stop. Like with the tigers, we stop completely because they say no more tigers going out, and we got no place to put them, so we stop. And then now we want to breed them again. The problem has came back. So that's the thing. Problem, I guess, with many of the species.
I think I agree with you. But the idea of distributing our genetic pool to other countries is also a good idea, as what we did recently. We sent our pair to Japan, in which they are successful in breeding our pair in bioparks. Yeah. Maybe Mr. Sanusi can add on any inputs on maybe on research.
Yeah, Japan, I think a lot of them, the zoos and as well as the universities, researchers also working on improving the reproduction of reproductive performances in the wildlife species.
Yes, yes, agree. Most of the research, especially on the behavior and on the breedings of Malayan tapirs come from Japan. Even we can even what you call it, collaborate with them. But although they have tapir, but it's not always from Malaysia. Most of it from Indonesia, I guess. So only one pair from Malaysia, but I don't think it's a problem in terms of collaborating for the good of the species. Yeah,
Yes,
I can see Dr. Lim. Yeah, maybe
Yeah.
you can have.
Yeah, I think I can give some input on that because I'm actually in Kyoto University right now. I used to work in I used to do my master in UPM. I work on genetic diversity in Malaysia.
Okay.
Now I'm in Kyoto University and I'm working on the same thing, but for the captive tapirs there. So far I can say that yeah, I know that were 2 tapirs sent to Japan in 2005, and then there were 2 calf bonds, right? And then there were also 3 individuals from Indonesia, actually. So far, Japan, they maintain a populations of about 31 to 33 usually, around that number. And I can see, because I've done the microsatellite analysis on them, also mitochondria, and I can see a lot of them are actually Thailand lineage, and it's actually quite not proportionate because I would say that about 20 something maybe is Thailand, and I mean 4 from Malaysia, and then maybe 3 from Indonesia, and also there were hybrids from Indonesia, so between individuals of Indonesian lineages and Thailand lineages, and then but that's an issue here as well because when I did this study, I raised the issue. Sorry, I raised the question of whether we should mix Indonesian and Malay Peninsula individuals in captive breeding because geographically we know they have been isolated for a long time, like 10,000 years, and also in terms of genetic as well, I found that there is some genetic differentiation as well. So, but the study is still ongoing, so I haven't come to a very solid conclusion yet. But yeah, so that's what is happening in Japan right now, that I'm working on the captive tapirs, and now I'm actually back in Malaysia for a month that I'm be working on the polyton samples using the new markers so that I can make better comparison between the Japan population and also the Malaysian samples. In the meantime, I'm actually also I'm actually contacting as well the Thailand, Dr. Wan Laya, also ESA and is A for the samples, but things hasn't been progressing very well because of the COVID situation. And Indonesia, I really hope if there's someone who can provide samples for us to do analysis, and then we can do a global comparison between individuals, in different regions. And then maybe that's where we can start to think about how we can plan the individual exchange and genetic exchange, or even some rare gene, we can preserve them better when we know. So probably that's what I can share for now.
Thank you, Dr. Lim. Dr. Sandie, maybe we can get some feedback from Biopark itself, Dr. Yoda. Are you with us, Dr. Yoda Takayuki?
Hi, I'm Takayuki. I'm glad to invite me to this conference. で、言いたいのが、あとお願いしたいんですけど、現在、4頭飼育していて、もう、うちのスペースが4頭でいっぱいいっぱい。
Currently, we have 4 tapirs under captivity, and it's really up to our capacities. Our capacity is full right now.
で、子供2頭をどうにか移動しないと次の繁殖ができないんですが、前回の繁殖から5年経ってしまっている。で、それを早く改善というか、新たな繁殖をして、次の繁殖、さらなる次の繁殖に続けたいんですけど、ちょっとその2頭の移動が決まってなくて、当館としては困っている。政治的な制約による。
So out of the 4, 2 are cubs, which we want to transport to other outside zoos. However, we are facing challenges because of some political constraints, and right now we're not yet able to transfer those 2 cubs outside of our facilities. And it's been 5 years since our last breeding, and we really wish to get cubs outside so we can go on with our new set of the next breeding. So regarding this political constraints, so we're not able to transport our 2 the baby or the child tapirs outside. Who can we talk to solve this problem?
Yeah, actually, I'm aware with the situations. However, yeah. However, I'm not now. I'm not directly involved with what you call it. Yeah, with the things. Yeah, yeah. But as we discussed before, yeah, Biopark could communicate with Malaysia through the Department of Wildlife and National Parks. Specifically to the zoological divisions under the Department of Wildlife and National Parks. Yeah, I think I can guide you to whom you should what you call it, talk with. Yeah. I think Dr. Yoda have my WhatsApp number. You can communicate with me after this. Yeah. Thank you, Dr. Yoda, for the kind inputs. Yeah. I think we need to get inputs from the zoo as well. Maybe.
Excuse me. One question, okay?
Sure.
5年間繁殖してないんですけど、オスとメスを一緒にすれば5年のブランクは消えますか？
So our question is, it's been 5 years since our last captive breeding. So if we put the male tapir and the female one together, will they be able to reproduce again?
You mean the adult Malayan Tapir?
大人の
そう
バッグですよね。Yes, yes, the adult tapirs. If we put the males and females together, will they be able to reproduce?
Yes, definitely they will be able to reproduce as long they are in good health. Yeah, but it may take some times, maybe 1 to 3 months. Maybe give them 3 months to what do you call it? To reproduce. Yeah.
Can I just add to that, Dr. Yoda? I mean, we see from the rhinos, the Smarten rhinos, unfortunately, that prolonged non-breeding, non-reproduction in the females in particular seems to result in a lot of cysts in the ovaries and very low reproductive capabilities, to the extent that they're actually infertile, and we haven't really found any wild tapirs that have been roadkill here in Malaysia and these kind of things that has any of those issues. So it seems like there's a good reproductive or healthy population here in Malaysia at least. But from the IASA and the Zoo Association, we have a few tapirs that have been phased out of the breeding program, and they're actually a lot the same kind of ovarian issues as the, what do you call it, the rhinos. So I would recommend you don't leave it too long. I think for 5 years, if they're still reasonably young, I think that should not be a problem. But don't leave it for too many years. That's my recommendation.
Thank you for your advice very much.
Thank you, Dr. Yoda. Dr. Sandie, maybe we can ask our counterpart in zoos. Yeah.
Yeah, Zoo Negara seems to have their hand up. Accidental or
hi. Hi, hello. Hi, Dr. Donny and Dr. Sandie and the rest of breakout rooms participants. I'm Junaidi from Zoo Negara, Malaysia, with my team from the education department. So I believe that it was a very fruitful discussion today. And then I'm so privileged to have involved in the team. So I just want to share some ideas and also some thoughts. My humble opinions would be, because we are from the education team. So for the zoo part, we have an important role to educate the youngsters and also to plan a lot of activities in terms of awareness. So I think I'm more touched on the SEPA program, which is the communication education and public awareness. So in the zoo, we have the facilities to run multiple programs for tapir conservation and also awareness. So we can joining for the tapir camp. Probably we believe that with the inspire of the conservation and also awareness among the audience, especially the young audience. So by using the zoo setup, which is we have the real experience with the Malayan tapir, whereby they can see from their eyes, their own eyes, to see the tapir and define what the features, what are important, what are the uniqueness of the tapir, especially our Malayan tapir. So we can also organize the tapir talk, the interactive with the tapir and also with the zookeeper. And at the same time, from there, we knowledge masters can appreciate more on the tapir. And then from there, we also get them know about the threats of the tapir, especially the roadkills and also the poaching and also the deforestation, especially, and the encouragement. We just want to open our facilities and also our services so that any agencies can tie up with the zoo so far in terms of education and also public awareness. So we can call the medias and also do some, probably we can assign 1 year for Year of the Tapir to give more impact in terms of physical event and also digital events. Yeah, thank you.
Thank you. From Zoo Negara, yeah. Yeah. I think the idea of having Year of the Tapir is very good. Something that we can think of. Yeah. Since we have the World Tapir Day, maybe we can extend it to the Year of the Tapir. Yeah, yeah. Maybe that something that we can discuss further. Yeah, maybe back to you, Dr. Sandie.
Back to me. Hey, Ju, you're still around in Zoo Negara. Good to see you. Many years. Okay, so we covered the public education part, the ex-situ part. We are talking ex-situ, of course. I think the biggest issue now it is the location to place these animals, as like Carl said. We can't leave them too long without breeding. How it is with the rhinos and all that. We have seen how disaster that is. Okay, the placement technologies. If there's a need, going for are we going for banking? Let's say the oocyte banking or the sperm banking. Donny? How you have something to say?
Yeah, well, first of all, the sperm or biobanking, personally, I would also suggest that one. I mean, you can never be too safe. The technology is there today. So I'll definitely propose that as one of them as well. The other thing, if it was up to me, I was in the university or in Pehilitan now, or in any of the regional authorities, I would also like to look at this having a crisis response team, like Dr. Donny mentioned. And I think one of those is also about behavior. So the female calf interaction, we are not really, that's probably one of the areas we are fairly blank in. You know, there are many types of species you can go in and actually boost the reproductive cycle by actually removing the calf. We just don't know whether that will work in tapirs. Most likely it will. But I think from a research point of view, that would be an interesting to do that. So that's another idea. I think, and then I have one more also, which is, I don't know if it has been done already, but if there's a genetic or DNA screening of all existing ex-situ individuals across, like Myanmar, those two they have there, in Thailand and Malaysia and in Indonesia, whether or not we have the genetic profile of all of them. If we don't, I suggest that is another fairly low-hanging fruit to start. So those are sort of few things I would think of. I just want to say I'm going to just pop into the other room. So it's not that I don't like you guys here.
All right, thank you, Dr. Kal.
Yeah. Thanks a lot, Carl. Hope to catch you in person. Okay, so we will collect blood samples on FTA, just that I don't know how good it is now, because I think some were, when it has fungus on it, I think that one is scrap really. So how have the department started to profile the tapirs, or are we planning to do that?
Yeah, for the Malayan tapirs, for genetic screening, for the tapir in captivity, not done yet, but we started with Malayan tiger. So we are doing genetic screening for all the captive Malayan tiger since last year. So probably we can extend the works for Malayan tapir, since we don't have much Malayan tapir in captivity. We only have around 40, so it's doable. I mean, we can be done. Yeah, and in fact, we can extend this what you call it, genetic screening for Malayan tapir within these regions, I think. Yeah.
Yep. So we need to get a working group set up, I guess, to get this done first.
Yeah, I agree with
it
you.
would require a lot of collaboration. Okay, that's one thing. Biobanking, is there a plan at the department? Because I'm sorry, I keep having to go back to the department because it's a protected species, so like nothing really much that we can do other than has all got come back to you.
Yeah, for biobanking, actually, recently the department get the recognitions of having the most biobanking wildlife specimen in our possessions. For Malayan tapir, yes, we are doing biobanking project for this species. This includes keeping their tissues, what else? Their gametes, yeah, and also their blood. We are doing it right now, yeah. But for semen collections, sorry, I'm not sure if Donny, you can hear me.
Yes, now we can hear you. Lost you.
Okay.
Wow.
Yeah. 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 Actually, we are doing the biobanking for Malayan tapir. We keep the tissues and also the gametes, but not really in active mode. Yeah, we are doing it when there are opportunity to do it. I mean, especially on gamete collections and cryopreservations. Yeah, but we are able to do semen. I mean, to collect the semen, and some are cryopreserved. Yeah.
Okay, preserve. But is it sure that it is the gametes preserve viable?
Unfortunately, most of the preserved gametes is not doing well. I mean, the percentage of live spermatozoa is not good. Yeah, I mean, after cryopreservations and after towing it, and we only can see a very small percentage of live spermatozoa. Maybe we need to optimize the extender and the way we we cryopreserve it. Yeah.
This is where we need more people to collaborate. That's all I can think of. Anybody else? Any other ideas?
Sorry, Mr. Sanusi is just beside me. He's talking something, but the microphone is not working.
Yeah, we can't hear.
Yeah, he's mentioning or he's suggesting to give opportunity to Dr. Rizwan to give some inputs, yeah, or anyone from Zoo Typing, yeah.
Okay, first of all, I would like to thank for adding me into this seminar. And then I was really interested in what Dr. Donny just said just now regarding cryopreservation of the gametes or the spermatozoa. I just wanted to ask, is there any efforts by Perhilitan to come to zoos around Malaysia to get the samples from the tapir that we have in our collection? So because in Taiping Zoo, we have 4 tapirs currently, and we have 1 young male. And I think he is a very good candidate for you to actually take sperm samples from semen sample from it. So is there any efforts from Perhilitan to come to any zoos to get samples from the tapirs itself? That's the only question I wanted to ask.
Yeah, actually, I would say the effort should come not only from Perhilitan, but it's a collaborative effort which involve zoo as well. So for the time being, we are focusing on Malayan tiger, which is the main priority of the department. But our team also doing the semen collections on other species as well, especially on endangered species like the gaur and also Malayan tapir. So maybe in near future we will do collaborations with zoos to samples to get the gametes from our Malayan tapir in all zoos. So at the same time, we will do the genetic screenings, so we can better manage the what you call it, the pairing of our Malayan tapir in captivity. Yeah, definitely we will involve every individual, especially vets in all zoos, to be part of the what you call it, programs.
All right. Okay. Just to respond, very open in terms of receiving any collaboration works from the department or any universities, because I think it's about time for us to actually have a sperm bank for each species that is on the brink of extinction. So just to inform Dr. Donny and the department, if you guys wanted to come, our zoo doors is always open for this type of research and conservation effort. Thank you very much.
Sure. Thank you, Dr. Rizwan.
Hi, this is Mark from UPM. I just have a question for Dr. Donny. You guys can hear me, huh? I just have a question from Dr. Donny in regards to the quality of the semen that was poor previously. What was the quality of the semen before thawing?
Thank you, Dr. Mark, for the questions. We did we did semen collections a few times since 2017. What we found that the quality of semen, it's about the normal spermatozoa is only 40%, 40%, yeah, 40 to 50%. Yeah, 40 to 50%. And in our recent, evaluations of the Siemens, which is, I think, last year or 2020, I cannot remember, the quality is dropped to 40%. But I don't think it is a major problem, big problem for the tapir, because the center still reproduce even with that quality of semen. Yeah, and maybe I would add the most common abnormalities on the sperm that we found is coiled tail and bend mid piece. Yeah. And I think this is opportunity for us. I mean, for the department and also university to study how to increase or improve the semen quality of the captive Malayan tapir. We are not sure about the quality of semen of Malayan tapir in other zoos, but in Sungai Dusun Wildlife Conservation Centers, it's only that what I mentioned. Yeah, but I think it's worth to do screening on other males in other zoos as well, just to compare. Yeah.
Just to ask a bit more, just now when you mentioned 40 to 50%, was that like the percentage that were actually alive or the percentage that were actually moving? Or would you know?
Yeah, the 40 to 50% is those who are normal. Yeah, those are normal. Yeah, yeah.
You mean these are the distant ejaculates that have 40 to 50 normal anatomy?
Yes. For the semen collection, we use electro-ejaculations technique. Yeah, but recently we have employed new techniques which are usually used in horses, which is called pharmacologically induced ejaculations, which not involve electro-ejaculators. So for that technique, the the percentage of normal spermatozoa is only 40%. Yeah, less than 50%. I'm not sure if it is. I mean, the technique of collection is give effect to the quality of the semen. That would to need to be studied lah. Yeah.
Yeah, I guess there's a lot of factors that are involved in terms of the quality of the semen. And so I guess this is just a response to what you mentioned just now when you see the post-cryopreservation, there's a lot of problems occurring. Right? Most probably because your initial sample was already so poor, so that's why your post-war samples, it becomes even worse.
If its life is only 40 to 50, after cryo, wow, you're lucky to get even 10%. Yeah.
Yes.
But they are breeding well. I mean, they're breeding, right? Even in Sungai Dusun.
Yes.
Maybe that's all they need. Or is it during the process something happened? I don't know.
Yes, the thing I think we need to study. Yeah, that's why we need collaborators from universities to study all those things.
Yeah. Riley, you have something to say?
Yeah. Okay, thank you. Okay. So far, in terms of, I just want to highlight again on the issues of the bank genetics. So just to inform that currently the zoo negara, we are having 5 Malayan tapirs in total in captivity, and then with one young male. So far, yes, I'm supporting Dr. Ridwan info that we are welcome to do any research being done. And in fact, Dr. Sandie, you can send a student to Zoo Negara, because currently we have been approached by the UPM student and the faculty, and also we would like to welcome from UMK also to do more research on not only for tapir but also on other wildlife. So we having that. You can contact me directly and the education team. And then back to the public awareness. So sorry guys, I'm more to education program. So far,
Can help. Anything helps.
so I believe that we have a lot of program being done as for now, because we know that our Malayan tiger is actually having crucial. So we don't want to have like the same with the Malayan tapir as well. So far, I'm personally joined the community awareness program in the rural area. So why not? We do some a similar program with the Malayan tapir, whereby we can go direct to the local community to support in terms of the awareness issues, especially on the roadkills and so on. So they actually can guard and also can take care of the tapir in situ and the wild habitat. So far, we can do a school outreach at the rural areas. And also at the same time, we can also engage the local community to support in terms of conserving the Malayan tapir and saving them. All right, thank you.
正規採取に話を戻したいんですが、
So can we again go back to the semen collection discussion?
牛の人工血などのような器具で、無麻酔で正規採取をしたことがありますか？無麻酔なし。
無麻酔。We have a question for cows using an artificial equipment. Have you ever collected semen on cow using such artificial equipment without using an anesthesia?
あともう一つ質問
So
で、
that's one question. We have a second question.
精液の希釈液、エクステンダーはどのようなものを使っているのか。
And what kind of extender are you using for the semen?
All right. I think I can give some feedback on the questions raised by Dr. Yoda Takayuki. So for the first questions, are we ever use artificial artificial vagina to collect semen from Malayan tapir? We never do artificial vagina to collect semen from Malayan tapir, but sorry, are you asking about using artificial vagina to collect semen from cow or from Malayan tapir? From cow or from Malayan tapir?
Tapir.
Okay, sure. Yeah, from cattle, yeah, we did artificial vagina to collect semen from cattle in Malaysia. But for Malayan tapir, unfortunately, we haven't did it yet because we couldn't. We probably it's probably difficult to train the tapir to mount on the what you call it, dummy female, or to mount on female which is not on estrus, but you can do standing sedations to collect semen from Malayan tapir, which involve giving some medications prior to the collections, prior to the semen collections. I have the protocol, and we in few occasions we managed to get semen with this technique. Actually, this technique is not really alien to species like donkey and horses, but these techniques were we did this first in Malayan tapir, and it working. So if you are interested to use the protocol, you can contact me. You can communicate with me to get the protocol. Yeah, but it involved giving medications called imipyramide. Yeah, 2 hours before the collections, and 15 minutes before the collection of semen collections, we give xylazine intravascularly, and the penis will drop, and we can massage the penis to collect the semen. Yeah, but not all tapir will generous enough to give their semen. Yeah, but most of the time we manage to get semen with this technique. Yeah.
Thank you. Please contact me with email.
Sure.
Thank you.
Dr. Zainal mentioned that we tried artificial vagina on rhinos.
Hello.
Yeah. Dr. Zainal.
Yeah, can you hear me?
Yeah. Can.
Okay, thank you. Sorry, line I'm in the wildlife reserve, so line is very erratic. I'm sorry about that. Well, we have used several times in the Sumatran rhino using the artificial vagina that was modified from horse AV, but the problem tends to be when the animal erects, the penis will start to get engorged, and there are two lateral projection on each side of the close to the glans penis. So this forms, and this, when it's fully engorged, it's really expanded almost 90 degrees of angle from the penis or from the glands. So it makes it very difficult, and some tried by modifying only applied at the area that is anterior to the lateral projection. So tapir, I know they also have the lateral projection, and this could be a problem using AV. Yes, you could condition them without any sedation. You could condition them to a shoot, and you can collect. We have tried it. You can get, but it's not very uncomfortable for them. That's my point. All right. Thank you.
Thank you, Dr. Zainal.
So, Dr. Zainal, since with your vast experience in getting semen samples, do you think there's any way we can get around it?
Well,
actively collect semen samples.
Yeah. After that, Dr. Sandie, we go for electroejaculation. There is I'm afraid there is Well, for us,
So
that is the best method to collect from the male rhino.
So wow. Okay, thanks, Dr. Zainal. So that means either Dr. Zainal, so that means electrodegradation. Animals have to be anesthetized or sedate or just anesthetized?
Full GA.
Yeah, right.
Full GA, yeah. But so far, I mean, the method is, it is safe. I mean, if you have the right right people and right anesthetics, there's so many protocol that they have done it, and I think we came across the 4-in-1. I can't remember exactly now.
Yes.
Yeah, 4-in-1, and it works very well. Not only for collecting semen, but also for ovum pickup in a female rhinos.
Okay, so we cannot escape.
I'm
All right.
afraid not. Yes.
Yeah, I'm sure the procedure should be rather safe to that. You need the manpower and the drugs. That's another thing. I think Malaysia sometimes we have issue getting drugs, but with our friends, I think no problem. We somehow manage.
Yes.
Okay. So I think that, Donny, is important that we sit down and form a working group. Even different working groups for different approach.
Yes, agree.
Nak letak mana pun satu hal. Where to put the tapir is no problem. Okay. What else? But so you talk about semen collection. You have done the ovum collection as well?
Yes, we did ovum OPU, but again, OPU, it requires, with tapir, again, everything requires, because these are not cows and horses where the anatomy and physiology is similar. So with tapir, probably you have to devise a method of OPU. But the one that was developed by IZW, transrectal, I suppose that would be the option. But with tapir, it has a long vagina, so again, you got to consider that. And there's the curve from the rear end. There's a slight curve to go in, and with the long, it has to be planned out. You can't use the normal OPU tools that you use for horses or for cows. So it has to be modified to sort of suit the species. And even for the rhinos, it took about 4 years to develop a tool that works well with transrectal OPU. So all this, I mean, you have to start now. I mean, we can't wait until it becomes a crisis, then we start. So whoever has research and grant or whatever, this should be an area to look at. It can even be a transcutaneous method going through the skin. And I mean, these are options that you guys can venture. And you have surplus tapir and you have surplus roadkills. These are where you can start off using roadkills as your sort of specimens to try and see the best approach to do OPU. And the other thing is that even with roadkill tapir, within 24 hours, the oocytes are still viable, the semen. So we shouldn't waste any of this. There's 160 over roadkills, and can imagine the amount of oocytes you can even at least mature and freeze them. These are for the future, for the next generation, not for us. But again, these are something that we need to do. That's my frank opinion.
Yeah, very true, Dr. Zainal. Thank you. So Donny, you said we have collected semen ovum.
We only managed to collect semen. We no attempt to collect ovum yet. Yeah. We may need Dr. Zainal to help us on OPU. Yeah.
So that's the live one, but because if we have roadkills, I mean, why not, man?
Yeah,
Covers
that
everything.
is the opportunity. Yeah.
And at the same time, we can study the anatomy. I mean, we can get the track out and then devise a modification for this species.
Agree,
Yeah, that's
yeah.
wildlife.
Yeah.
Every single one has something different.
Yeah, actually, we already have one publications on the anatomical structure of Malayan tapir female Malayan tapir organs, but in this case, maybe we need to redo it. In fact, to what we call it, to find the best tools to do the OPU. Yeah. Dr. Sandie, I think Dr. Lim is raises her hand.
Please.
Yeah, thanks. Sorry, just wanted to add to what Dr. Zainal said just now. Actually, I've been thinking like, not just semen or the ovary, but also like, you know, gut, looking into gut microbiomes, right, into their fecal samples, into what they have eaten in the past. I don't know, maybe before, for example, for roadkill samples, we can look into even the gut, the food composition, even the gut microbiome, and also RNA samples to look into the functional genes and all that, and especially like, for example, East Coast and West Coast, the different habitats. So I'm just wondering, because I feel like we have a lot of opportunities, especially in the past 1 year. We know that there were like 24, maybe 23 tapirs died in a row, but I'm not sure, is there any like what do you call it, like efforts to organize effort to collect this kind of samples, like among the office among different states, like how we can work together to collect this kind of samples. Like if there's something happen, then we can collect and we can send it to the for example, WGRB in Pelitan, in Cheras, this kind of stuff. I'm just wondering.
Yeah, Dr. Lim, I think Dr. Khairul Nizam can answer that questions. I think he is around. He is the head sections of research in Perhilitan. Maybe Mr. Kai, can you give some input? Are you around?
He's in the group.
Yeah, he's in the group. I'm not sure. Maybe he's away from the laptop. Yeah. Yeah. Never mind. Actually, our research team in Perlitan actually doing the collections of biomaterials, includes those that mentioned by Dr. Lim. Yeah, and it's not only done by the team from the headquarters, but also involve the staff in states and also in district. Yeah, and not only involve Malayan tapir, but all species that available in Malaysia, especially those endangered and threatened. Yeah. I mean, those are listed in the WCS Act 2010.
All right, thank you. So it's actually possible. Let's say if I submit a proposal right now, it's possible to access this sample for doing some research.
Yeah.
Are they available?
Yes. Yeah, actually, the samples in the lab in the National Wildlife yeah, Wildlife NWFL forensic lab is available for researcher to do research. Yeah, the only thing you need to do is to prepare a proposal and send to the Director General of Perhilitan. Yeah, of course you need probably need a collaborator. I mean, the representative from Perhilitan as well. Yeah, to do the work together with you. Yeah.
Yeah, And one more question to Dr. Donny, actually, because since you've been working on the tapir as a vet, so I wonder, like, what is your, do you have any like main concerns about like health and welfare of tapirs in ex-situ institutions? Are there any things like or diseases that you're looking into that you hope that can be probably solved using genetic tools or like as in the word mentioned, genetic screening?
Yeah, I think via genetics, via genetics or DNA, maybe you can help us to improve the pairing of our Malayan tapir in captivity. Yeah, physically they probably will accept each other, but some of our pair is not what you call it. Is not reproducing. I mean, they not produce babies. We could look into their genetics on
olfactory cues.
Yes. Yes, Etheline?
Yes. Oh no, like olfactory cues, something what do you call, like a sense, smells or hormones or hormonal cues.
Can you repeat?
Sorry. Yeah. I mean, it's like not just like, I mean, it's linked to genetic as well, but like olfactory cue, like smells or hormonal cues or something like that. Because I remember like Dr. Gita had done something similar in the past. But I'm not sure if anyone is still doing that.
Okay. Actually, I'm doing hormonal study on our captive Malayan tapir, but not only hormonal study, but I incorporate with others techniques for the estrus cycle monitoring of our Malayan tapir. I try to understand their estrus cycle in captivity. Yeah.
All right.
Yeah,
Yeah,
maybe
I'm
for
more-
further informations or discussion, maybe you can communicate with me. Yeah.
Okay.
Yeah. For the, I mean, for specifically for the research. Yeah.
Yeah. I am very interested in looking into like a genetic compatibility between like a captive bred captive breeding pairs, like a genetic compatibility in terms of genes, in terms of immunology, this kind of stuff.
Yeah, I think that will be a very good what could call it, very good research. Will help the department and also the zoos in terms of pairing of the male and female. Yeah.
All right. Thanks so much for.
Can I, sorry, just to interrupt, can I just add something to this? I mean, in IASA, when we connect to individuals, we actually have what we call psychological profiles of the individual. And of course, there's no doubt that that's an expression from a genetic composition too. So it'd be nice if we could look into something like that. I mean, if we have a very dominating female and a very timid male, it usually doesn't really work out that way. So in a way, you can say animals need to fall in love also. There is some sort of personality clashes very often. So yeah, it would be very interesting to see if you can find the genetics of that as well. That would make everything easier.
Thank you, Dr. Carl.
All right, we only have a few more minutes to go to wrap up. So we come back to the microbiome. So far, we think that the nutrition that we give to the captive animals is sufficient and good enough, I guess, because they are breeding. Just an interesting fact, when I was when we was at the HQ, we have a collection of the skulls. The weight of the captive animals and the weight of the roadkills, there was quite a significant difference. I don't know why. Is it because of the beef we feed them? So whether does that have any indication to their reproductive or their movement or anything else, we are not sure. But if we are doing the microbiome of the gut, I guess it will help when you can even identify what is, well, there's already a study, like what is their diet. But again, getting what their diet in the wild and what we can provide in captivity is another issue as well. So how do we need to present it later on or something like that?
Yes, the idea is one of you would just go through the different points of sort of proposals that we have made in this group.
So Mark is writing out, is it?
Yes.
Yeah, later I'll pass to Dr. Donny.
Sandie, can I add one point, one last point? See, when
you can add as many points you like.
All right, thank you. Number one is that whatever we plan, there are resources that is very limited. And say if we are looking into semen, do we have a lab? I'm sure we have a lab that can accommodate and can freeze the semen. But when we talk about oocytes, I doubt we have a lab that has a CO2 incubator that is working to mature the oocytes before you freeze or before IVF. So these are things that we can talk about it, but we need on all angles. The resources has to be there. The person doing it has to be there. And I do not know whether we have those. And we can go on planning for years, but if we don't have that and we are not focused, we're not going to get anywhere. And we keep losing oocytes. And I'm sure with the females that got killed in road kills, there are many oocytes that you could retrieve, as we have seen with other ungulates as well. So these are things that also need to be considered when we present. We not only present a plan, but it has to have the criteria of a good plan. And I also agree with you on the tapir that we examined previously in terms of nutrition. Just by, I think it's a work from Copenhagen or from I guess so. They're doing a radiograph of the mandible, and you could see even the skull. You could see the huge difference in terms of those in the wild and of those in captivity. So maybe again, this is something that we need to consider. Are we overfeeding the tapir with energy food or with fat, or is that going to affect breeding, or is that affecting them? So these are a few things that we need to sort of bring it up again in your discussion or presentation. Thank you.
All right, thanks, Doc.
Hi, Dr. Zainal. In regards to the CO2 incubator, actually UPM has one, but it just hasn't really been used so much. But if there's a need, you can look into that.
That's good to know, Dr. Mark. Thanks.
Yeah, Mark
Yep.
is in the production. Yeah, you're right. So resources from all over. In fact, not even when we breed, if we are successful in breeding so many tapirs, just to put them is already an issue. Like many zoos are already have reached their capacity or they are over the capacity, just like how we were with the Malayan tigers decades ago.
Yes, you're right, and that's why I suggested in Sabah we have a space for 10 tapir in captivity.
Okay.
Yes, and just to note, we got 30 oocytes from the Bonian bunting that was that came in that was short and all that. We managed to salvage 30 oocytes, but because the CO2 incubator was not functioning, we lost all 30 oocytes. Just to let you know the gravity of having equipments.
You mean the oocyte has to be in the incubator?
Yes, you have to mature them.
Or mature them first, okay.
Yeah.
Before you store in.
Yeah, before you do the IVF or you store them. Yeah.
Okay, yeah, that will be an issue.
Just a note.
All right. So obviously we need a team of people with all these knowledge to list down what we need to have first before when we start.
Sorry, just one thing that I would like to share about ovary cryopreservation. I do know one colleague. I'm not sure about who are the experts in Malaysia, but I do know one colleague from Kyoto University, and then she has been doing this ovary cryopreservation for many animals around Japan. So usually the Japan zoos will send the dead animals ovary to her, and then she will extract the ovary, and then she will preserve it. And I think they're building an ovary bank or something like that. So just information. If anyone need her information, can reach out to me.
Okay, thanks, Salim. Yes. What you want me to have a closing remark or what?
Yes, just a wrap up. Yeah.
So Mark, how? You have the points or something? So we talked about the facilities we have, location, number of animals that we have, and the potential of using advanced technology to further improve on the reproductive performances of these animals. But of course, we need to evaluate the animals, do a national, at least a nationwide profiling of the animals that we have. If we can expand it to the regional, that will be even better. We need collaboration internally and with fellow collaborators outside the country as well. Yes. And coming back to resources, road queues management. So Donny, you mentioned that even the state officers, they would be able to reserve the road queues, but we have to meet the minimum now. So what? I just leave with it. But what about, are they able to collect the gametes? If they can collect the organs, I think they're quite capable, and to retrieve is one thing, and to store is another issue. So I'm not sure whether our state also, or you have a team to dispatch immediately, like, okay, tapir is over there and we have to go and collect daily or something like that.
Yeah, most of the time our vets will help to collect the the organs, the reproductive organs, and bring back to our laboratory for further process. Yeah.
So it will last long enough, I mean, for you to preserve the gametes? Just asking now.
Yeah, we did get some samples. I mean, testicle samples from few species, including tapir. But for tapir, unfortunately, the organs, I mean, the testicle from Malayan tapir that we collected within 24 hours, when we process it, the semen's already died. I mean, no life spermatozoa. But for wild boar, yeah, after 3 days, we still can get live spermatozoa from testicle samples. Yeah.
Okay.
Probably get a protocol. I mean, just build up a protocol even for vets collecting it. There must be a protocol, kan? Even for ovaries should last at least a day or so.
Yes.
So I think just protocol, I think.
Yeah, agree with you, Dr. Zainal.
Yeah.
So how is it? We wait to for them to close the breakout room, or we have to leave? How this message we have to go back to the main room? Do we just leave this room and it will go back?
Yes,
Yes, all participants can leave the breakout room and go to the main room right now. Thank you.
Okay, thanks for everyone's participation. I appreciate your feedback. So see you back in the main room. Thank you. Bye.
Thank you, Dr. Sandie.